like show you with the data, like how it will look, how indel will look like, what will happen in heterozygosity, what will happen in homozygosity, and all those things he'll be talking to you in detail with real data. Okay, whatever he has done in his lab. So so you will get an exciting opportunity. But before that, it is mandatory that you know the principle of Sanger sequencing, and you understand the nitty-gritties of the chromatograms which the Sanger sequencing generates, right? So that is why initially we saw the machine, like how it works, what are the different components. Okay, and now all of you know it works on the dideoxy principle, and the phosphodiester bonds are the key. So here we have the phosphodiester bonds, and this OH is the key for let's say extending the chain. But when this is deprived of the hydrogen, of the oxygen, it becomes only hydrogen remains here. So already this is a deoxy position, there is a hydrogen here.
Online we have
Acha, okay, fine. Okay, fine. So I did like my regular class. Okay, so this is the diester bond. Okay, so we need to have a OH here so that the chain can be extended. But we, in addition to DNTPs, we add the DDNTPs also here. And then that is one. It has 2-3 principles. But immediately that comes to our mind. If you are asked in an exam, you'll always say Sanger means dideoxy, chain termination method, those kind of stuff. But very then the almost 99 out of 100 students will say that, or 100 out of 100 students. But then the another important step is there, that is called cycle PCR. Okay, that is a very important component in DNA sequencing. Okay, so now I'll talk about the cycle PCR also. And because cycle PCR uses generally any PCR means 2 primers, forward and reverse primers. You being students of molecular biology or genetics and that kind of background, you know, like in case of RAPDs and ISSRs, and there are some other marker systems where we use a single primer. But there also, though we use a single primer, they will work as both forward and reverse primer. But in case of cycle sequencing, it is not like that. There is forward means forward, reverse means reverse. Okay, so that is the difference between cycle sequencing and those systems which use just one primer. Okay, that is one thing. Because if you look at the context, when you are doing RAPD and ISSR, you are taking the entire genomic DNA as the template, right? So then there are so many matching or complementary sequences there, so that's why they can go and bind wherever complementarity is there. So it can work as both forward and reverse. But in case of this cycle PCR or the sequencing ke liye, whatever PCR we do, in that case your template is already very well defined template. You know, like this is the gene you want to sequence, and this is the place you want to examine. So you are already, your template is extremely well defined, and in that case how your primer can go and work as both forward and reverse, that is not possible. So that is why only one site is there, either forward site is there or reverse site is there. So you will be adding only one primer there. So this is one difference. And then whenever you add this kind, when you do this kind of sequencing, like there is a limited template, okay, and you add only one primer, either forward or reverse at a time. So when you do that, you the basic idea of the PCR of 2 power n, that is the basic idea of PCR. So that 2 power n is now no more there. That is exponentially now it doesn't work. Now this whatever amplification, this is an arithmetic what to say, increase is happening, not the exponential increase is happening. So the amplification is arithmetic. So this is yet another difference, okay, between these two. So generally when you give the samples, you will be just giving the PCR products. So the PCR products, let us say, like you will be only doing this step, and depending upon like 500 to 800, this is the normal size of the you will be deciding, because most of the capillary electrophoresis DNA sequencing systems will have the 37 capillary centimeter long capillaries. So that is that cannot go beyond a good quality sequencing data of 800 base pairs. So that is why we also restrict this here. If we use like a 50 centimeter capillary, then we can go for in up to 1.2 kb also. But for that 1.2 kb, we are not very sure what is the capillary wherever the vendor you are giving, or some big institute you are giving, whether they are using which capillary. Every time we cannot go and ask everyone. So it is safe. Like you design primers only of this size, okay, 500 to 800 base pair. two smaller like products also, like the sequencing does not happen properly. It is difficult to standardize the DNA sequencing protocol for very smaller amplicons, and the larger amplicons anyway will not work. See, basically this is only a capillary electrophoresis, means only what, like very small size also it can segregate. But at the same time, you just imagine like what is the difference between like 100 base pair molecular weight and 101 molecular weight. Okay, now you think what is the 1 kb's molecular weight and 1001's molecular weight. Now just think of the difference. That itself will tell you anything going beyond this limit cannot be resolved properly. Okay, this is the main reason why a machine cannot resolve beyond 800 base pairs properly. Okay, because the what to say, arithmetic increase between 1000 and 1001 is different from what is 100 and 101 for the capillary to run and resolve it. After that, everything gets like kind of cluttered. After that, it cannot like it's not able to pull it that much, so that you can see that also well. See, yeah, then there could be some cases where you are like determining the sequence. In case of genome editing, the idea is little different. You want to see where the whether the mutation has happened, and if happened, where it has happened, and what kind of mutation has happened. This is what you want to design. But sometimes before you, let us say, start the program, you want to know the entire gene sequence, okay, or the cDNA sequence, so that you want to design guide RNA. So here in that step, you can even you can design anything in such a way that overlapping primers, they can after forward reverse align. After that, you can even get 1.2 kb good quality data. So you can reduce the number of overlapping primers and do that. But in case of genome editing results, you should not do any such compromise. Okay, because you have a targeted region, and if you are having a too long a region, multiple guide RNAs design, then you design those many PCRs reactions. Okay, so that is the good thing. Anyway, Dr. Solanke will be talking about that in detail. For you, like I would like you to just remember, you please restrict yourself here. Okay, if you are giving for Sanger sequencing. Generally what happens, you send these PCR products, and sometimes you may clone, okay, PGMT clone karke PCR product koi you may elute clone, and then you can give. Anything is okay, it is up to us. If you want to resolve homozygosity, heterozygosity issues very clearly, then in that case you can clone also and send also. So generally we restrict ourselves here, but what happens after that? Okay, this is only for you to understand, nothing more than that. Now whatever PCR product when you give, it is in a tube, and it will have some unused primers, unused nucleotides, everything. They will hinder with the further cycle PCR as well as with the sequencing. So it is very, very important that first we clean them. So that is why this is the most popular cleaning method, exosap based. Exo stands for exonuclease. So any primer, the 3', 5', it will go on the exonuclease, will degrade them, and then the shrimp alkaline phosphatase, this will remove the phosphate group, so they are no more DNTPs. They are now nucleosides. So now they cannot go and attach themselves and become a proper nucleic acid at all. So that way also we took that capability away, that capability from them. Now whatever is remaining, you can simply do ethanol precipitation, and you can use that. Okay. Fine. So this exosap after this cleaning, for of course for these enzymes to work, you have to incubate them, and after that we go for the cycle PCR, or this is called, another name is sequencing PCR. So whatever things I said now, that exponential and linear amplification instead of exponential amplification, using a single primer only at a time. Okay, if you want to do the it is recommended that for genome editing also you do the forward also, you do the reverse also separately, and get both of them sequenced, because I don't know whether you have learnt tilling. In tilling also, like this kind of will tell you, like let us say if it is a 300th base pair is the place where this editing has happened. Now